Hello everyone, this is Lecture 2C, the third and final lecture for Week 2 of Applied Immunology. In this session, we'll add to our previous discussions on innate immunity by introducing two additional processes which promote innate responses, and these are programmed cell death and the signaling cascade called the complement system. In the last lecture, we covered pattern recognition receptors, or PRRs, and learned how their activation by pathogens triggers signaling cascades which help initiate immune responses. In addition to PRR signaling, infected cells can also engage another form of innate immune defense, which is programmed cell death, abbreviated as PCD. I want to emphasize that these are forms of cellular suicide that are controlled by the activation of intracellular signaling cascades, hence why we refer to them as programmed cell death. So this isn't uncontrolled cell lysis. Now, if you consider a cell that is infected with an intracellular pathogen such as a virus, the targeted killing of that cell is obviously beneficial for host defense in that it removes the survival niche for that pathogen, which helps kill the pathogen as well as release antigens that can be used to instruct adaptive immunity. Interestingly, there are actually a few different ways in which a cell can undergo PCD, and we'll introduce three primary forms of PCD in today's lecture. First, cells can undergo apoptosis, a form of PCD that all of you have probably heard of in your introductory biology classes. Apoptosis is morphologically characterized by the dissolution of a cell into several plasma membrane-bound vesicles called apoptotic bodies, which kind of look like this cluster of little bubbles. Apoptosis occurs following activation of a family of cysteine proteases called caspases, although the pro-apoptotic caspases are different than the caspase 1 that we introduced in the last lecture when we talked about inflammasome activation. Caspase 3 and 7 are shared executioner caspases of apoptosis, though other caspases are involved with upstream signaling as well. Notably, although apoptosis can be triggered by stimuli associated with infection, it is also used under homeostatic conditions, such as during development, when old or unneeded cells need to be eliminated without stimulating an immune response. So apoptosis can therefore function as a non-inflammatory form of PCD that is actually tolerogenic, meaning that it doesn't stimulate innate or adaptive immunity. Second, there's pyroptosis. Pyroptosis occurs downstream of inflammasome activation, such as NLRP3, AIM2, or NLRC4, which we just talked about. Last week, we learned that inflammasomes converge on the cleavage and activation of caspase 1, which in addition to processing the mature forms of IL-1 family cytokines, can also cleave and activate a protein called gasdermin D. Now, gasdermin D forms pores that insert into the plasma membrane. So the result of this is that the cell swells and bursts. So this is a lytic form of PCD. Because pyroptosis releases cell-associated molecules that can be inflammatory, as well as IL-1 family cytokines, this is viewed as a pretty highly inflammatory form of PCD. The last form of PCD we'll discuss is necroptosis, which occurs following the activation of the RIP kinases, RIPK1 and RIPK3. As kinases, these proteins then go on to phosphorylate target proteins, including something called MLKL, which functions in an analogous fashion to gasdermin D, in that it assembles pores in host membranes that lead to cell swelling and rupture. Notably, the RIP kinases also have some transcriptional activity, as necroptotic cells exhibit a burst of inflammatory chemokine and cytokine production, which accompanies cell lysis. For these reasons, necroptosis is also viewed as a highly inflammatory form of PCD. Considering how cells undergoing PCD can produce many different types of inflammatory signals, these forms of cellular suicide make up an important additional layer of innate immunity, as they allow a cell to sacrifice itself in order to generate either pro-inflammatory signals, as is the case with pyroptosis or necroptosis, or anti-inflammatory signals, as can be the case with apoptosis. Again, any form of PCD succeeds in removing the replicative niche for intracellular pathogens, which also allows for the release of cellular antigens, including pathogen-associated antigens, to stimulate adaptive immunity. Furthermore, specific forms of PCD offer fine-tuning of the immune response mounted against dying cells, which depends on the specific cytokines and chemokines produced by dying cells, as well as the release of some inflammatory molecules called damage-associated molecular patterns, or DAMPs, which can function similarly to PAMPs. And this occurs because they can bind to pattern recognition receptors, although we won't discuss these any further in this course. Lastly, I want to stress the fact that PCD can be initiated by signs of cellular damage and stress. For apoptosis, this includes signaling through the TNF receptor, as well as molecules released by stressed mitochondria. For pyroptosis, this involves the diverse stimuli that can trigger NLRP3 activation, which we discussed in the last lecture. And for necroptosis, this includes both the TNF receptor as well as some other cytosolic sensors of viral infection. So PCD also differs from most forms of pattern recognition in that it can be triggered independently of PAMP recognition by PRRs, and instead occurs in response to more general signs that cellular integrity has been compromised and the cell needs to be eliminated. Switching gears, let's start with the second topic of today's lecture, which is the complement system. Complement is another innate immune signaling cascade, and this is responsible for recognizing pathogen surfaces and then marking them in a way that flags them for destruction while promoting local inflammation. There are 3 different pathways used to activate complement: the lectin pathway, the classical pathway, and the alternative pathway. All 3 of these differ in their initiation steps, but converge on the shared goal of inducing the enzymatic cascade of complement activation here in the middle. Regardless of the upstream initiation pathways, complement activation leads to the same 3 outcomes listed here on the bottom: inflammation, which is done through the production of inflammatory mediators that help support innate immune activation and recruitment, the opsonization of pathogens, which is a process that promotes their phagocytosis, and targeted pathogen lysis, which is accomplished through pore formation in pathogen membranes. In addition to that simplified overview schematic, let's start by focusing on some of the basics of the complement cascade. This is really just a signaling cascade of proteolytic enzymes and their substrates. All of the molecular components of complement are produced by the liver, but they circulate throughout the body so that they can be activated in response to local infection in pretty much any peripheral tissue. All complement enzymes exist as in an inactive state until pathogens are detected, in which case the signaling is rapidly amplified as enzymes are activated sequentially following the initial recognition of the surface of a pathogen. Lastly, complement is a really crucial mechanism for defense, primarily against bacterial and fungal infections. So this is a very important innate immune signaling pathway. Now let's get into some of the reasons why complement can be difficult to learn and remember. First, the signaling components are named using a pretty arcane nomenclature system, and this can be really annoying because it has numerous exceptions to the general rules that we try to establish. So let's go over some of these rules. The first is that proteins are named by a C paired with a number, and cleaved fragments of each protein are denoted by a lowercase letter. So in the case of the protein C4, which is the full-length protein, proteolysis yields C4a and C4b fragments. This is fine and pretty straightforward when you're just considering a single protein, but it can get pretty unwieldy when referring to multi-protein complexes made up of multiple fragments such as C4b2a, C4b2a3b, etc. A second rule is that the proteins in the alternative pathway are named using an uppercase letter, while again the cleaved fragments are denoted by a lowercase letter. So in the case of protein B, which is the full-length protein, proteolysis yields a BA and a BB fragment. A third general rule is that usually small soluble fragments of proteins are denoted with a lowercase a, while the larger fragments that are bound to a pathogen surface are denoted by a lowercase b. However, one important exception to this is C2, which is cleaved into C2a and C2b. The last general rule is that the numbering of complement components is usually done in order of their enzymatic activation, except for C4, which is out of order. So C1 actually activates C4, which then activates C3, then C5, then C6, 7, and 8, and then eventually C9. Now, all of these things make complement a little unwieldy to remember, so memorizing the pathway is kind of seen as a hazing ritual or a rite of passage with immunology students. But since the exams for this course are open note and open book, you don't need to really strictly memorize these pathways. I still expect you to understand the key functions of the complement pathway, but you won't be getting any nasty exam questions asking about obscure complement signaling complexes. We'll still cover the signaling pathways, but again, I want to de-emphasize rote memorization here and have you focus more on broader conceptual understanding of the complement cascade. Let's begin with the lectin and classical pathways of complement initiation. The lectin pathway is triggered by attachment of soluble factors to the surface of a microbe. And these soluble factors are either mannose-binding lectin, abbreviated as MBLs, or phycolins, both of which recognize sugars with molecular modifications that are only found on the surface of microbes and not host cells. The classical pathway is triggered by a similarly shaped molecule called C1, which can bind either directly to C-reactive proteins or CRPs on the pathogen surface, or to antibody pathogen complexes. There are conserved aspects of the lectin and classical pathways. One is that the binding domains of these soluble factors essentially function as PRRs, and that they bind to pathogen-specific surface molecules. This is true for MBLs, phycolin, or CRP-bound C1. Number two is that the MBLs, phycolins, and C1 all look structurally similar, which we can see in the diagrams here on the left. They have a college and stem, which is then connected to recognition domains, which are the clusters of balls on the end of the molecule that actually bind to the pathogen. These soluble molecules have homologous enzyme domains as well, as the MASP1 and 2 found in lectins is homologous to C1R and C1S in the classical C1 protein. Now, it's important to note that from here on out, the lectin and classical pathways are completely identical in terms of what happens downstream of these initial binding events, so they really only differ in terms of how the cascade is initially triggered. The next steps involve the catalysis of other complement proteins to initiate the enzymatic cascade portion of complement. These diagrams outline the activation of the lectin pathway, but keep in mind that the classical pathway is the exact same concept, except it's initiated by C1 binding to the pathogen surface. In both pathways, the next step is to cleave the protein C4 in order to generate soluble C4a and deposit C4b onto the pathogen surface, where it seeds complement complex formation. In the lectin pathway, this is accomplished through the enzymatic activity of MASP2, while in the classical pathway, this is mediated by C1s. Once C4b is deposited, it can then cleave and activate C2, which establishes a complex on the pathogen surface called C4b2a. This is a C3 convertase, which simply means that it will cleave and activate the next protein in the cascade, which is C3. This has two important downstream effects. One is that the pathogen surface gets decorated with a whole bunch of C3b molecules, and this is a process called opsonization, which is relevant for the phagocytosis of this pathogen. And the second is that a C4b 2a3b complex is formed, which is a C5 convertase that allows for the cleavage and activation of C5, which is the next protein in the complement cascade. We're going to quickly switch over to the alternative pathway, which differs from the classical and lectin pathways in how it forms a C5 convertase. Now, in the alternative pathway, there's no pattern recognition binding with a soluble factor recognizing sugars on the microbial surface. Instead, C3 can actually undergo spontaneous hydrolysis, where it can then bind to a protein called factor B. This binding recruits another protein called factor D, which cleaves B into the subunits BA and BB. This complex then functions as a C3 convertase, which cleaves additional C3 molecules into C3A and C3B. Again, as in the classical and lectin pathways, C3A is soluble while the C3B subunits attach all over the pathogen surface and opsonize it. Once C3b is seated on the pathogen, factor B and factor D will bind, leading to the cleavage of factor B into BA, which is soluble and floats away, and BB, which stays with C3b on the pathogen. This complex then recruits a second molecules of C3b, creating a complex called C3b2BB, which is a C5 convertase that allows for the processing of C5. Now, I know that this is a lot of jargon up until this point, but all the complement pathways converge on C5 proteolysis at the pathogen surface. The lectin and classical pathways accomplish this through the establishment of the C4b2a3b C5 convertase, while the alternative pathway does this through its C5 convertase, which is C3b2BB. In either case, C5 is recruited and cleaved into soluble C5a and pathogen-bound C5b. This processing of C5 is critical for the three main outputs of complement signaling. C5a is the most potent soluble inflammatory signal produced by complement and is essential for the inflammation induced by complement. C5a is also required for the phagocytosis of C3b-coated microbes, which is related to the opsonization and phagocytosis outcome. Lastly, C5b, attached to the pathogen surface, enables these cells to be targeted for pore formation and subsequent lysis, which kills the pathogen. So we'll next discuss how complement signaling achieves these three primary outcomes in greater detail. First, the production of soluble complement products, that is, the A fragments produced following proteolysis, promote inflammation by activating the vascular endothelium. As we discussed in Lecture 2a, this activation leads to two key outcomes. One is an increase in vascular permeability, which allows for the egress of fluids and other soluble molecules into an infected tissue where complement activation has occurred. The second is chemoattraction and induction of adhesion molecule expression by vascular endothelium, which promotes leukocyte extravasation from the vasculature into the infected tissue through diapedesis, which we learned about earlier this week. Second, the opsonization of pathogens through complement effectively tags them for phagocytosis. Opsonization refers to the decoration of the pathogen surface with C3b following C3 convertase activity, which occurs with all three complement pathways. There are two things that need to happen in order for opsonized pathogens to be successfully phagocytosed. The first is that C3b binds to complement receptors that are expressed on the surface of phagocytes, such as this one here, CR1. The second is that the soluble factor C5a is detected by the phagocyte by binding to a C5a receptor expressed by phagocytes. Importantly, both of these receptor-ligand interactions must occur through a process known as coincidence detection in order for phagocytosis to be engaged. So an opsonized pathogen can't be eaten up in the absence of soluble C5a, and C5a receptor activation cannot make a phagocyte eat a non-opsonized pathogen. And this coincidence detection provides an additional layer of regulation to the phagocytosis outcome of complement signaling. Third, C5b attachment to the pathogen surface triggers pore formation through its cell membrane. This is referred to as the membrane attack complex, or MAC. Notably, there are no more proteolysis events in this portion of the complement cascade, only binding interactions and eventually pore formation. So this all occurs when C5b binds to the next three members of the pathway, which are C6, C7, and C8, forming a protein oligomer that inserts itself into the cell membrane. Collectively, this complex is referred to as the motor of MAC pore assembly. This oligomer then recruits C9, which is a subunit of the complement pore. So several C9 molecules then assemble together to form a pore that pokes a hole in the pathogen cell membrane. You can actually see these MAC pores via electron microscopy images here on the bottom left on the surface of a pathogen that's targeted by complement. These pores function by disrupting ion homeostasis, which results in an influx of extracellular fluid that basically pops the target cell and kills it. I'll end this lecture by driving home the importance of regulating complement signaling so that it only acts on foreign invader cells and not host cells. This is accomplished through two primary mechanisms, the first of which we already touched on when discussing the lectin and classical complement pathways. So in these cases, the soluble factors that initiate those pathways, either MBLs, phycolins, and C1, basically function as pattern recognition receptors that they only bind to foreign molecular motifs that are not found on self cells. The second mechanism is that host cells express a variety of molecules that function by displacing, blocking, or otherwise inactivating complement proteins in case they accidentally target host cells. Some examples of this include the expression of a C1 inhibitor, which is secreted by host cells and will remove the enzymatic subunits C1R and C1S from the C1 complex. This essentially inactivates it so that it cannot cleave its substrate C4. Another good example is the host cell expression of a plasma membrane protein called CD59, and this functions by binding to the membrane attack complex at the stage of C5b678 formation. So CD59 binding to this complex blocks the recruitment of C9, and therefore MAC pore formation is prevented. There are several other negative regulators of complement that can prevent accidental targeting of host cells, but I wanted to introduce these two briefly today so that you can appreciate the backup mechanisms that we have in place to ensure that this powerful method of innate immunity is really only deployed against pathogens. This has been a pretty dense lecture in terms of protein complex naming and vocabulary, so here's a summary slide outlining the differences and similarities between the lectin, classical, and alternative pathways of complement activation. We can compare these three pathways in terms of their initiation events, as well as their enzymatic cascades and the complexes used to process key substrates such as C5, as well as their shared final outcomes that help promote host defense from primarily bacteria and fungi. In summary, we've learned more about both programmed cell death and complement activation as additional mechanisms of innate immunity, and these help supplement innate immune activation through pattern recognition receptors. Programmed cell death has a number of important functions in promoting host defense, and distinct forms of PCD differ with respect to their signaling requirements as well as their downstream effects on immune responses. Notably, PCD can be triggered by stimuli other than classical PAMPs and can therefore alert the immune system to the presence of compromised cells that must be removed. Next, the complement system is a proteolysis cascade that can be activated by 3 separate pathways, all of which converge on the assembly of C3 and C5 convertases, which enable the generation of inflammation, the phagocytosis of opsonized pathogens, and the lysis of pathogens through pore formation. This concludes the lectures for week 2 of Applied Immunology. You have a reading assignment to accompany these lectures, which is outlined in the syllabus, as well as an extra credit weekly quiz and a discussion board prompt. This week, you'll be responsible for providing a brief summary of an innate immune signaling deficiency disorder, so please see Canvas for additional details on that. Good luck with all of your assignments, and I'll see you next week for our second module on innate immunity.